Okay, so by this point, you should have created and activated your free DNA Subway account. And in this video, I'll walk us through how to create a new project and then add data to that. So if we look at our instructions here, what we need to do is log into DNA Subway using what we just used. We'll start a purple project. We're going to use paired end reads. That's a type of high throughput sequencing data we'll talk more about later. And then we're going to name our project. So let's go into DNA Subway. The top left panel here is where you log in. So you enter your username and password. And then what we want to do is create a new project. So we'll go down to meta barcoding analysis, which is what we're doing. Click that. For the options here, we want to do paired end reads. That's the only file format we have. And then for our project title, so let's see, I said to name it Bio 178 Week 2 Microbiome plus your initials. So I have an example here. My initials are LG, so that's what I would call it. So I would enter that for my project name. I think I did that twice. Okay, so Bio 178, Week 2, Microbiome, LG. And then for our description, you don't have to put exactly what I put, but I suggest something like project created for Week 2, Bio 178 lab on whatever date it was. Putting dates is good. It will help you keep track of if you need to do a new project, start over. The dates will help you keep track of everything. Once we have that entered, click continue. And now you are in your new DNA Subway project. So there are, they call them stops because it's like Subway, DNA Subway. So there are three main stops here: metadata and QC, clustering, and alpha beta diversity. Today, we're just going to be focusing on the three different components of this first stop here, metadata and QC. So if you look, it kind of guides you through what you should be doing. So right now, the only option that we're allowed to click, so if you try to click somewhere else where there's this red X to it, it says it's blocked, so you can't do that. So basically, it'll tell you what you need to do in order to proceed on with the steps. So the first thing we need to do, if it has a little R next to it, that means you should be running that one. So the first thing we need to do is manage our data. So click Manage Data. And then for FASTQ files, which are the high throughput sequencing data files, FASTQ is the type of file that comes off of the high throughput sequencer. For this particular week, we are going to be using example data files that were provided to me through a workshop I did this summer. It's a really cool data set about looking at the different bacterial communities present in male and female garter snakes. So we're going to click this option, Add from Cybers. So we want to add data files that are already present within Cybers. You want to go to Shared Data. And this pathway, it gets a little confusing, but we'll just walk through step by step how to find this data. So if we scroll down, now we are finding the data. So we want to go to the shared data, which we just did. iPlant DNA Subway. Got to scroll down. iPlant DNA Subway. Click that. And then we want meta barcoding data and fall 19 snakes. And then these are three different types, three different locations on the garter snakes that they took samples from. We are working with musk, so the musk gland on the snake. So click musk. And now you're going to see a bunch of different files pop up. And it's really important in the instructions here, it says to click all files except a couple. So female for musk2 and male to musk2, we do not want to add. When I was doing this a couple days ago before I was writing it up for you, I ran into some issues, so I worked them out, but it's really important that we do not add these two files. So we're basically clicking everything except female for musk2. So if we go and click through, I realize this is tedious, I apologize. Okay, so female for musk2, we do not want to click. So we'll skip that one, keep going through for all the females, and then for males, we do not want male to musk2. So we'll click. We don't want male to musk2, we'll skip that one. Click all the other male files. So we're basically clicking everything with a .fastq.gz file extension, except those two I just clicked. Just those two that I skipped. And then at the end, there's this other file. We're going to use that in the next step, so don't click that one either. So once you've clicked all of them except male to musk2 and female for musk2. Let me make sure those are the right ones. Okay, then what you want to do is add selected files. That's 38 files, so it'll take a little while to process. We'll get this little message here. While we're waiting for that, we can look at the instructions for the next step. So we've added the FASTQ files, and the next thing we need to add is called a metadata file. This is essentially telling DNA Subway which of those files we just added, what sampling group they belong to. So the main thing we're looking at here is males versus females. And so this metadata file is really important to tell DNA Subway how to analyze the data. It's still processing, so I'm gonna, oh, there we go. Okay, so now we have all our FASTQ files there. If you scroll down, for metadata files, we're gonna also add this from Cyverse. You can just click this last location folder, and it'll take you to that last folder we were just in. Scroll down to the bottom, click Musk Metadata, add selected files. And then because we didn't add those two files I told you to skip, we need to do this extra step of editing our metadata file. So go to edit and it'll pop up. This is actually what that metadata file looks like. And so what we did is female for musk2 is one of those ones that we didn't add. So we don't want that listed here because we don't actually have that sample. So what you can do is right click to remove that row. And then we also want to do the same thing for male to musk2 and remove that row. And then we save that. Make sure you save it, otherwise it won't keep those changes you made. Now we want to go back, scroll down to the bottom, and now we can click validate. So it's saying no errors were found, everything matches as it should, and now we can click run. And so now you can see that this demultiplex option now has a green R next to it. So we're now ready to do that step, which I will cover in the next video.